In recent years, stem cell research has received significant interest in the fields of science and medicine. Stem cells are extraordinary, with the capacity for self-renewal and differentiation into diverse cell types such as intestinal and adipose cells. This process occurs through several mechanisms, including cell division and the activation of specific signaling pathways. Environmental factors also play a crucial role in the regulation of stem cell renewal. These factors can include both external and internal cues, such as growth factors, hormones, physical forces, and cell-to-cell interactions. Humans are regularly exposed to environmental pollutants and toxins such as endocrine disruptors, which can cause abnormal stem cell behavior and limit the potential for renewal. One common endocrine disruptor found to affect normal stem cell behavior is bisphenol A, or BPA. BPA is found commonly in sports equipment and food packaging, as well as personal care products. One impact of BPA on stem cell renewal is its role in upregulating the production of adipose cells, limiting the ability of adipose-derived stem cells to differentiate into other cell types critical for the structure and function of the human body. BPA is structurally similar to estrogen, and as a result, it has the affinity to bind to membrane-associated estrogen receptors, or ER. BPA activates estrogen receptors A and B, disrupting normal estrogen signaling. This results in an upregulation of transcription factors involved in adipogenesis, or the formation of fat storage cells from stem cells. Insulin-regulated growth factor IGF-1 controls the mass of adipose tissue by regulating adipogenesis, but binding of BPA to ER simulates the binding of IGF-1 to its receptor. This in turn upregulates the phosphorylation of CEBPs through the phosphoinositide 3 kinase pathway. Activated CEBP-alpha induces the expression of PPAR gamma 2, a key factor that directs the differentiation of adipocytes by regulating the formation of the functional transcription factor that binds to peroxisome proliferator response elements PPRE in DNA. This results in increased accumulation of triglycerides and increases the differentiation rate of adipose-derived stem cells into mature adipocytes. This process reduces stem cell renewal potential as it decreases the number of undifferentiated stem cells available for differentiation into other cell types such as chondrocytes, myocytes, osteoblasts, and neurocytes, which are critical to the growth and development of cartilage, bone, and nerve cells. Thus, it is evident that BPA can affect stem cell renewal as well as normal functioning and maintenance of the human body. However, similar effects can be found in diet and nutrition as well.
In mammalian cells, a calorie-restricted diet can promote intestinal stem cell self-renewal via intracellular and intercellular signaling. A calorie-restricted diet means taking in less calories than habitually while receiving all essential nutrients and avoiding malnutrition. In intestinal infoldings called crypts, Paneth cells and intestinal stem cells are located directly adjacent to one another, and they communicate. When Paneth cells experience caloric restriction, ATP levels decrease, thereby increasing AMP levels. AMP binds to AMPK subunits. When LKB1 phosphorylates the AMPK activation loop, AMPK activates and phosphorylates Raptor. Raptor blocks mTORC1 kinase from phosphorylating its substrates, which in turn increases the rate of expression of an ectoenzyme called BST1, which converts NAD+ to cADPR. cADPR is the signal that is sent from Paneth cells to intestinal stem cells. When intestinal stem cells receive cADPR, they respond by increasing calcium ion release, which activates CAMKK. CAMKK then phosphorylates AMPK. Activated AMPK increases the expression of the Nampt gene, which increases levels of NAD+, activating the expression of sirtuin-1. Sirtuin-1 promotes fatty acid oxidation and deacetylates S6K1, which makes it easier for activated mTORC1 kinase to phosphorylate it. After S6K1 is phosphorylated, intestinal stem cell self-renewal is promoted.
Understanding the mechanisms and environmental factors that regulate stem cell renewal can contribute to improving therapeutic strategies and developing innovative treatments for a wide range of diseases. However, continued research in this field is crucial for unlocking the full potential of stem cells. Thank you for listening.